Hello, welcome again. And this is the second part of our lecture about digestion and the digestive system. Now, let's get into the more complex process that is the one performed by herbivores and carnivores. Let me put this in slideshow. All right. What are the main difference, or what are the main also characteristic of herbivores? Well, they present, talking about teeth, they present incisors and molars, but there are no canines because, well, if you think about it, there's no need to actually hold a prey that wants to run away once you got it in your mouth. And at the same time, you don't want to tear any flesh, because these, of course, are animals that are being fed on, or they feed on plants, on vegetables. So what they actually need, they need molars and premolars, which are going to be the more important teeth, for cropping and grinding the cellulose of plant, which actually is the main component of plant cell walls. And by grinding thoroughly that cellulose of the plants will actually allow to increase the surface area for the bacteria to be able to digest that cellulose and hemicellulose and break it down through the enzymes into glucose. One characteristic of the of herbivores, in particular, for example, cattle and deer, they don't have upper incisors. They only have incisors in the jaw, not in the maxilla, and they are substituted by a pad in the mouth. So this is important for you to know. So they will be characterized by presenting 4 stomachs. They are also called ruminants. And the 4 stomachs are the reticulum, which is the first stomach, smaller than the rumen, and is normally the compartment where all the heavy objects are going to fall when the ruminant is ingesting its food. Then followed by a rumen, normally formed by a dorsal sac and a ventral sac, followed by a omasum, and the omasum is a kind of round-shaped stomach that is on the right side of a rumen, which function is still being debated. It's not very clear. The omasum actually looks like a book with plies that actually resemble the pages in a book, and still not clear again what's its main role in digestion, but it has been theorized or suggested that probably its main function is to help grinding the feed, the plants in this case, and also it's important for water absorption. And finally, we have the abomasum, also referred as the glandular stomach, or sometimes called the true stomach. So in the rumen, the reticulum, abomasum, we will find the bacteria and protozoa that actually populate these 4 stomachs, and they actually produce the digestion through fermentation, through the fermentative process. So they will be harboring anaerobic bacteria and protozoa to break down cellulose. That's another concept that is important in ruminants. Ruminants, actually, they have inside their 4 stomachs a moist environment, a warm environment, and very low oxygen, almost in anaerobic condition, which is the conditions that the bacteria and the protozoa need for fulfilling its fermentation process. The other thing is animals do not produce cellulase or the enzymes that actually break down cellulose and hemicellulose. So it's only bacteria that are able to do so. So this is an important concept. So actually, the ones responsible for breaking down the cell walls of plants are actually the bacteria and the protozoa. And then ruminants also present quite a large rumen cecum that also harbors symbiotic bacteria that will contribute to the fermentation process. So these ruminal bacteria and protozoa, they will produce what we refer as volatile fatty acid, which is acetic, propionic, and butyric acid, which are going to be absorbed through the ruminal wall. And the other characteristic of these ruminal bacteria is that they are actually able to synthesize protein from a non-proteic source of nitrogen. So, for example, in ruminant, in livestock, it is common to feed them with a certain percentage of urea. So the bacteria in the rumen will actually utilize this urea, which of course, urea is not a protein source of nitrogen, to produce microbial protein. And this microbial protein is normally of very high biological value, meaning that it's easily digested and absorbed by the animal. So the actually ruminant, what is actually feeding itself is feeding out of their bacteria that it's harboring on its four stomachs. So they also present a long, small, and large intestines. And ruminants are characterized by taking frequent meals, normally hundreds of kilograms per day of wet matter or as fed. And there is always a constant throughput in the digestive tract. That is part of the physiology of the animal. And also, the other thing I wanted to refer briefly here is that ruminants are able to fulfill rumination, which is a process where they actually perform a controlled vomiting. So this ruminal content will be pushed back to the mouth normally while the animal is resting, and it will be keep on chewing by the ruminant, let's say the deer or the cattle, and the excess of water is going to be reabsorbed. And this is done to increase and keep on grinding and making sure that the whole plant and structure is really broken down, so that will allow the bacteria and the protozoa to digest through fermentation these vegetable components. So here we have a diagram showing the anatomy more clearly. We have the esophagus. So food will move from the esophagus into the reticulum and the rumen, and from there it will go through the omasum, as we said, and normally will fulfill a role in water absorption, follow in the transit by the abomasum or true stomach, sometimes referred as well as the glandular stomach, which is actually secrete chlorhydric acid and pepsinogen, pretty much similar like monogastrics. So we can say that that region of the digestive tract or ruminant towards the anus is very, very similar to monogastric, with the difference that normally cattle and ruminants present a large cecum and column as well, which is not the case as for example, a carnivore. This is a slide I like very much because it's an electron microscopy picture image taking of the granules of starch being digested by bacteria and protozoa, that you can see that they are actually on the surface of a starch, and you can see that some of the granules have already been digested, because you can see kind of holes being made on its surface, on its structure. So if we look at the digestive anatomy of a horse, the horse is an animal that actually we can place them midway between a ruminant and a monogastric. Of course, they are herbivores as well, but they don't have a rumen, right? They don't have four stomachs, but they have only one glandular stomach, normally not very big in size in proportion to the rest of the digestive tract, but they are characterized by presenting a very big, a large column and a very big cecum as well. The cecum is normally placed on the right side of the abdominal cavity. I forgot to tell you this. The rumen normally is located more towards the left of the body cavity in ruminants. So in the case of bloating or bloat, that means the excessive accumulation of gas produced by fermentation, which is, of course, it is pathological situation. It's not normal, but sometimes they have this problem. You will see that the flank that is swollen is going to be the left flank. Most of the absorption will occur in the small intestine, but the absorption of volatile fatty acids and some vitamins, which actually is produced in both the cecum and column of a horse, will be absorbed. If we think about it, almost all animals actually have some type of fermentation, normally in the cecum. Even humans, for example, we have a cecum and we have symbiotic bacteria as well. So we are able, but of course, very limited to digest some fiber content of the feed. In the case of hindgut fermenters like the horse, of course, they have a much greater capacity of cecum and column, as we can see in the diagram, and they actually are able to sustain their life based only on fermentation products from the cecum and the column. One thing also I forgot to mention is that ruminal microbes and the microbes present in the cecum of column of herbivores are able to produce most of the B12 complex or B complex vitamins and vitamin K, which is important. And also, it is one of the deficiencies normally present in vegetarian diets. So we see another diagram showing the large proportion or the large size in proportion with the rest of the digestive tract of the column and the cecum of a horse. Now, let's have a quick look at the small intestine. Well, the nutrient is, we can say that is a region of the digestive tract that is characterized by having a great absorption properties, where most of the nutrients are going to be absorbed and utilized by the animal. And it's very interesting how absorption is actually increased in the animal, how they do actually increase their absorption capacity. Well, first of all, by presenting a long intestine, and it's going to be folded many times. Just to give you an example, some mammals' intestine actually exceeds 8 times their body length. Okay, that's give you an idea of how long the intestine is. And of course, making a longer tube folded many times inside a body cavity is a way of increasing its absorption capacity. But it's not only that microstructure, we can say, structure of a microanatomy, which could be considered a folding intestine, but they also present the villi, which are actually folds inside the structure of intestine that increases the capacity of absorption of the intestine. But the intestine also present on the surface of the epithelial cells a brush border called microvilli that will actually increase the surface area of the intestine 1,000 times, so 1,000 times capacity. So you can see there in the diagram, we have the villi, which will be the bigger folds or the bigger structures. And then, of course, it's going to be lined by the epithelial, intestinal epithelium, and each cell will present this microvilli. And so it's a structure under a structure and inside another structure, and all of them actually aiming to increase the absorptive area of and metabolic area and digestive area of the intestine. That's another diagram showing you the structure of intestine, and you can see there also that the vilus actually present this kind of finger size or finger shape, and they are inside. You will find the capillaries that actually are going to absorb the nutrients, and also the lymphatic vessels that are characterized by having a blind end, and they also look like a finger projection within. And other animals that are not vertebrates, like the annelid, like the earthworm we saw in the lab, actually present another structure called the typhlozoal, which also destined or aim to increase the absorption capacity of the intestine. Is like an inner fold of the gut of the earthworm. When we look at canines and at the carnivores in this case, we see the presence of canines and incisors, but fewer molars. Why? Because they don't actually need to grind any plants or any cell walls or any cellulose. But I mean, most of the cases, canines normally swallow large portions of feed without much mastication. And normally they are characterized by taking 1 meal per day, or sometimes even 1 meal every few days. So the teeth and the mouth of a carnivore is actually adapted and developed for killing, holding the prey, and tearing the flesh or the meat. And they present normally a small stomach, and they are called monogastric because that's the only stomach they have. And this stomach has, although the capacity to stretch to accommodate large meals. And if the animal keeps on eating larger meals, this stomach is going to be able to stretch more and more and adapt to that kind of more frequent feeding. They are characterized by presenting a much shorter intestine compared with the herbivores, and they normally produce, they have a reduced or no cecum present, and when present sometimes it's just a vestige like the appendix in humans, okay? It's a vestigial organ. They normally take in frequent meals, on average 1 meal per week, or much longer even intervals. And just to give an idea, you know, there are several research has been done and shows that the best feeding interval for dogs, for example, domestic dog, is once a day for adults. Of course, the puppies, of course, need to be fed more frequently. Other animals, like the birds, present other kind of specialized digestive organs, and that's the case of the crop, where they actually are able to storage food temporarily. And some birds actually secrete what we call the crop milk, which is kind of a special enzymes and secretion that sometimes they use to feed their chickens, their chicks. And it's actually like a stomach. And then we have the gizzard, which is a chamber of the stomach of bird that actually is capable of pulverizing or grinding food. So it forms what we call the old pellet, and sometimes it includes stones that are actually being picked up by the bird and ingested, and that helps in the grinding process of the feed. And that's typically found in those animals or organisms that have no teeth for chewing. So it's kind of a substitute of the chewing process that takes place in the mouth of those animals that have teeth. So we have here the gizzard with the presence of the gizzard stones. Okay, let's have a look now at the general mechanisms of digestion. As we mentioned before, it's a hydrolytic process, meaning that the chemical bonds of biomolecules are broken down by hydrolysis with the help of a molecule of water. Again, that will help to free the energy contained in the chemical bonds, because that's the way we store the energy. So for example, this general digestion equation we can refer to, we use it as an example, the sugars that with the presence of water and digestive enzyme, could be an amylase, for example, or disaccharides, will actually break down the sugars with the help of the water molecules in green and blue you can see there. For example, we can have sucrose, which actually is formed, as you know, by glucose and fructose that bonded together with the help of an enzyme, or the enzymes, which in most cases enzymes are proteins. So the substrate will bind to the enzyme and will form what we call an enzyme substrate complex that with the help of water will actually hydrolyze and break down this glucose fructose bond and break that bond, and we will have one molecule of glucose and one of fructose ready to be absorbed as monosaccharides. Because as we remember, we always move in digestion from a macromolecule, a more complex molecule, towards the more simple molecules to help in absorption, for them to be ready to be absorbed easily by the cells of the digestive tract. And the products will be released and the enzyme will be free and ready to bind to other substrates. That's another concept important, is that normally enzymes are not wasted in the reaction, so they can be utilized many times in more enzymatic processes. That's another diagram showing the hydrolysis, in this case of peptide bonds, not in sugars. So the the polypeptides or the protein will be broken down on its structural units or amino acids, and amino acids normally will be absorbed in the digestive through the digestive tract by the with the help of active transporter, specific transporter for specific amino acids. So how do enzymes work? Well, you having a kind of a refresh or review from chemistry. So you have the enzyme, which have a very specific active site where only the specific substrate can actually attach to it. So the substrate will attach to the enzyme, be broken down, forming this enzyme substrate complex with the help of water. So that's why it is called a hydrolysis process, producing the product or the breakdown product, and the enzyme will dissociate with the product or the substrate and be ready to take part in another reaction. So the biosynthesis is normally referred as constructive anabolism. And on the other side, when we move towards catabolism or digestion, we see breaking down into the component of the substrate is called the destructive process. So what is the main role of enzymes? Well, enzymes, what they do is they act by lowering what we call the activation energy necessary for pushing forward the reaction. So without the presence of enzyme, normally we will need a much higher energy to actually activate this enzyme substrate complex to take place and the digestion to be done, right? And another concept that is really important to make clear here is that enzymes actually do not provide energy to a reaction. So the only one that can do that is ATP. Remember, I always mention it to you, I call it the energy currency of the body. So, and the other thing that is important to know, and that explains why such a regionalization or compartmentalization of the digestive tract, enzymes are very specific in the range of pH they become active. According to the enzyme is the pH. So we see that, for example, we will find a whole gradient of pH in the digestive tract that goes from a value of 1, normally present in hydrochloric acid, stomach acid, for example, and to pH of, for example, of 5, between 5 and 6, that is characteristic of a bile acids, and urine and saliva normally work under alkaline conditions. So that's a pH between 6 to 7 or neutral. So this acid production of chlorhydric acid is really important in the stomach because it is under this low pH that pepsinogen is one of the most important enzymes. Actually, it's a proteolytic enzyme, will become active as pepsin and be ready to break down protein. But this actually conversion of a proenzyme, pepsinogen to pepsin, only occurs under acidic conditions, and we're talking about between 1 to 2. And here we see this more clearly, this kind of pH range, that how it varies according to the different compartment of the digestive tract we consider. So we move from the saliva, which has a pH of 6.5, and saliva contains in some species, mainly, for example, in the human, amylase and bicarbonate. So bicarbonate is a buffer that actually helps controlling the pH. Then when we move towards the stomach, we'll find the gastric juice, which has a very low pH. We're talking about 1.5, but so low, so acidic, that is necessary for the stomach to secrete mucus that is going to line the whole internal surface of the organ and protect the mucosa, stomach mucosa, from being digested by its own juices, its own hypochloric acid. So normally in the gastric juice, we will find pepsinogen, chlorhydric acid, renin in ruminants and mammals. Renin is an enzyme that is very important for lactating mammals because it's actually the enzyme that coagulates or clots the protein casein containing milk. So that's very important for milk digestion in the young animal. Then we have the bile, which is has a alkali alkali pH between 7 to 8, from neutral to alkali. So that's the characteristic of bile salts and pigments and also cholesterol. Then as we move towards the posterior part of the digestive tract, we get close to the pancreas. The pancreatic juice will have a pH between 7 to 8. And of course, the pancreatic juice is the main load of enzymes necessary for digestion, where we will find trypsin, chymotrypsin, carboxypeptidases, all proteolytic enzymes, which are going to kind of continue. And that's the other concept. One enzyme in one region will continue the digestive process initiated for the previous region and the previous enzyme in the digestive tract, moving, of course, from the mouth to the anus. So we have, again, the trypsin, chymotrypsin, and carboxypeptidase, which are proteolytic enzyme breaking down proteins to more basic structure like amino acid to be absorbed. Then we have lipases, which are going to be digesting lipids. Amylase, like starch and disaccharides. Nucleases, DNA and RNA, and bicarbonate, again, present as a buffer. And remember, a buffer is a substance that actually contributes to control the pH in the digestive tract. Then we have, finally, other membrane enzymes present on the surface of mainly of epithelial cells, like aminopeptidases, maltase, lactase, and sucrase, which are all disaccharides, will be breaking down disaccharides. Then we have alkaline phosphatase, will be breaking down phosphate components. And then we have nucleotidase and nucleosidase, also digesting DNA and RNA. And it's not only an enzymatic process, but also it is a mechanical process that contributes to, and it's important for, think about the food on the gut, on the intestine. So it is important for this muscular contraction in the intestine to keep on mixing all the time the enzyme with the food particles, enabled to be having a more efficient digestion. So we have what we call a segmentation, which actually helps to break the feed and the helps in the enzyme action. And then the peristalsis, which is kind of a continuous muscular contraction in kind of waves through the intestine, along the length of the intestine, pushing the food to the posterior part. And so as it moves forward, you can think about a kind of conveyor belt. As it moves backward, so the whole pH changes, and as the boluses gets into the different regions, different enzyme will pour down and will start the action, which is going to be continue on the following sectors or compartments of the digestive tract. And this is a nice diagram I like, because it actually shows how the pH changes as we move in ruminants, in this case the cattle. We start with the pH of 8 to 8.4 in the mouth, followed by the rumen that has a pH between 6.5 to 7, so close to neutral. Sometimes, due to grain overload, and we'll see in another slide, it can happen that this pH can go really, really low, like 5, killing some of the ruminant microbes, and important production of acetic acid, which actually produce a metabolic acidosis and a lot of problems in the animal, sometimes even death. Then we have the reticulum with a pH of 6, or mesum 4 to 5. So we can see that as we get closer to the abomination of the true, as we call, glandular stomach, where chlorhydric acid is produced in the ruminant, we have that pH goes down to 2 to 3. And then the small intestine is more towards alkalinity, because of course we have the bile and the pancreatic juice that contribute with bicarbonate. So pH will go up to 6.7 to 8, and the large intestine a pH between 7 to 8. So we have the rumen. And this is the scenario where in the ruminant we have, for example, we are feeding too many grains, too much grain. So we're producing a lot of starch is being getting into the rumen. So it will be rapidly digested by the microbes in the rumen. And that will produce a lot of acetic acid, decreasing the ruminant pH. And then we see that the microflora start having problems surviving in such an acid environment. And then we will have the developing by the animal of a subacute, what we call subacute ruminal acidosis. Milk fat is going to be reduced as a consequence of that metabolic problem. But also we will see that we have other problems like laminitis or feed problems in the animals. And sometimes some of the bacteria die, actually freeing endotoxins due to the fact that they are dying, which are going to be absorbed through the walls of the rumen. And that will actually contribute to worsen to the problem of acidosis in the animal. And of course, when we reach a point when we have a pH of 4 within the rumen, we have the acute symptoms of acidosis, animal death. And of course, the microflora is not able anymore to digest the fiber from the plants. Here is a very curious case of animal that I wanted to mention because, of course, in zoology it's good to compare different animals and different digestive systems. And bees and wasps are able to produce formic acid for their defense, like stings and jets. And actually, there is one animal in particular, which is the giant anteater, which they cannot produce stomach acid, so they are not able to produce chlorhydric acid in their own stomach. So they actually need this formic acid from their prey for digestion. And that's the reason why the giant anteater actually what it does is it needs to eat ants to get this chlorhydric acid from their actual prey. So they utilize the formic acid from the ant diet for their own protein digestion. Remember, the acid condition of the stomach, it's necessary for the conversion of pepsinogen into pepsin. So we can distinguish three main pH zones in the digestive tract of monogastric. So we start with the mouth with a pH slightly above 7, close to alkaline condition, we can say. That is good for the activity of amylase, for example. And when we move towards the stomach, we have a pH of 1.6 and 2.4 between that range, which is very acidic. And of course, it is fundamental, the closure made or performed by the sphincters, the esophageal sphincter or in the cardiac region of the stomach, and the pyloric sphincter, which actually prevents the very acid content of the stomach to get either into the esophagus or into the duodenum. And in some cases, just as an anecdote for you to know, heartburn, sometimes we might suffer of that. Normally happens when the cardiac sphincter doesn't close very well. So part of the acidic content of the stomach gets into the esophagus and actually giving us this kind of burning feeling, which is called the heartburn. And the small intestine, of course, we're moving towards a more alkaline environment around 7.5. This is due mainly for the buffers and the alkaline secretion from the bile in the liver and the pancreatic juice, which has bicarbonate as a buffer, increasing the pH in that region. So we have then the biomolecules will go through the process again of hydrolysis, and according to the nutrient we are talking about, will be the type of breakdown that is going to happen. So we start with complex carbohydrate. We start with the polysaccharide, of which the digestion will start in the mouth with the amylase in the salivary gland, actually degrading or starting to degrade polysaccharide, mainly starch, to produce smaller polysaccharides. In the stomach, there is no digestion or enzymatic action for sugars, but it will continue in the small intestine. So these we're going to be converted, the polysaccharides into disaccharides and into monosaccharides by carbohydrases. And these monosaccharides are going to be absorbed through the intestinal lining. In the case of a protein, the main digestion of protein will take place within the stomach through the action of pepsin acting on more complex protein, forming or producing small polypeptides, which are going to be broken down into peptides, and through the action of trypsin and chymotrypsin, which actually will continue the action of pepsin will form smaller peptides, which under the action of peptidases will break them down into amino acids, which are, remember I told you, they are absorbed by the intestinal cell through specialized transporters and very specific transporter. Sometimes some transporters actually carry one only specific amino acid, not another one. Then in the case of a fat, because of its composition, is a much complex digestive process. So normally we have the formation of fat globules thanks to the bile, which is going to emulsify. Remember, fat and water do not mix very well. For this reason, the bile salts, what they do, they produce what we call micelles, or very small, tiny drops of fats, which is going to contribute to the digestion through lipases, and they're going to break down into monoglycerides and fatty acids, which are going to be absorbed through the intestinal cells. And these triglycerides are going to be, these fatty acids and glycerol are going to be re-esterified into triglycerides and form chylomicrons or kilomicrons, which are going to be absorbed through the intestinal villi and are going to get into the lacteal, which is the name that the lymphatic capillaries receive, and going to be carried as kilomicrons to the different tissues in the body to be utilized. And finally, nucleic acids are going to be mainly digested in the small intestine by nucleases, forming nucleotides, which also be digested by nucleosidases, and will form the more simple components, which are the nitrogenous bases, sugars, into sugars and phosphates. Now, let's have a quick look at some zoological interesting situation, which is the feeding and digestion in the snakes. So snakes are actually able to eat meals that are about 150% greater than their own body mass, which is kind of unique. If you think about it, some jokes about this to you to see. Anyway, so what are the main challenges that eating a large meal implies? Well, first of all, to capture and overcome a prey that is several times bigger than you. The second challenge will be to swallow that prey. And the third one is the immobility that actually happens after eating because of this. The whole metabolism of the animal is going to be directed to digestion. And normally they are immobile during that time. And of course, that makes them very, we can say, susceptible to be captured by their own predators. And of course, digest and assimilate this vast amount of nutrients without causing a digestive overload or a body overload, organic overload. So how they actually subdue and control their prey? Well, in some cases, some snakes, they crush or they entangle, they suffocate the animal by cutting their circulation and by asphyxiation. But some other snakes actually use a different mechanism, which is the venom or poison. So we have the case of the rattlesnake, for example, and they have these venom and associated teeth, which are normally curved. They are actually, some people compare them with syringes, hypodermic syringes, and approximately half of all the snake species carry some type of venom. This normally venom is a concoction or a cocktail of about 25 different enzymes, mainly hemotoxin and myotoxin and some neurotoxins. And they are used, this poison is actually injected by the snake to the prey to immobilize them, and then they will start to, this enzyme, digesting the prey from the inside out through the action of the venom or the poison. So giving a head start to digestion. So it's a way of saving some energy there as well. And then the second challenge, remember we said, so we solved the problem of mobilizing a prey that is several times bigger than you. So it can be by strangling or asphyxiation or injecting a venom. Then comes the other challenge, which was the swallowing. So they actually can do that because they have a kinetic skull. We can say, we call it kinetic because it can actually open many times its original size. So by a kinetic skull with ligaments, the quadrate and dentary ligaments. So you can see the bones there, the quadrate, and the dentaries that are able to open that jaw really, really big. And that allows them to swallow something as big as an ostrich egg, like you can see that small egg snake is swallowing. And the other thing is, of course, presenting a skin that can be stretched several times. So that, of course, adapts to swallowing on the throat region. But the other thing is they normally have their teeth facing backwards, and that's important. So they are in an angle, so it's not an obstacle for swallowing whatever they are swallowing, their prey. And the other thing is they have strong throat muscles, and also they present an anterior glottis. So that's important, so the animal doesn't suffocate, and they can keep on breathing while they are swallowing a huge prey. So how do actually they need to protect themselves from predators? So they do that by actually keeping immobile, inactive, and they normally conceal in bushes, for example, so the predators cannot see them, that they are actually in the digestion process, that normally they are more susceptible to be attacked, and there's nothing they can do. The other thing is they regurgitate the rest of the meal that they haven't digested. And I'm going to show you this because it's very interesting, which is the case of the pythons. And they actually, they are able to, and we can see in this transmission electron micrograph that actually show you the rapid postprandial means after meal, lengthening of the microvilli of the python's intestinal cells. So they reach a peak in length by day 3 after meal or post-feeding. And we can see that after the digestion is complete around the day 6, the microvilli actually go back to the original length. So you can see a whole adaptation of the whole of the digestive tract of this animal for actually having huge meals. So we have there as well, we can see images of a small intestinal of a similar size Burmese pythons during fasting. So they didn't have any meal on the top picture. And as we move downward, 2 days post-feeding and 10 days post-feeding, going back to its normal shape and size. But you can see on day 2, the intestine has increased in diameter. And that is due primarily by the hypertrophy of the epithelial cell microvilli that we saw before as a response to these huge meals that these snakes are able to do. And last, I want to talk a little bit about cooking. Cooking is the process of cooking meat. Actually, what it does, it gelatinizes the collagen, and that makes the protein easier to be digested by the animal or by the human, by the person. Since cooking breaks down, is kind of a breaking down these different structures of the feed before being ingesting the food. So the body is actually able to absorb more calories. And in the case of raw food, of course, it can be digested as well, but those calories will be mainly not gonna be very useful for the host, will be used more by the bacteria in the digestive tract. So cooking is actually it's a process that begins the breakdown of the molecules by adding heat, mainly protein breakdown, that increases the net energy yield by the animal that is ingesting the food by 30%. So that is fuel energy demands for the brain that fulfills that. So raw food diet is rich in vitamin and micronutrients, but bear in mind that it will not yield enough energy, so that will lead to weight loss. So with this slide, I finished the digestive system lecture. Thank you very much for your attention.